This is a presentation of the University of Wisconsin-Parkside.
I'll start by saying this is I guess I've been part of the lab group in Milwaukee for about 3 years now. I came from a postdoc and I'm in a sort of in-between position. They call me a visiting professor for whatever that means. Even though I'm there, I don't visit from anywhere else. But so we've been working on this system for about 3 years, me personally. And I come from more of the microbial ecology, lake sciences background. And the person I'm working with, Sandra McCullen, comes more from a human health, pollution tracking sort of background. So this talk is a little bit of the merging of those two backgrounds together. This, I guess, is the first time that I've sort of given this talk in a broad overview. So I'm leaving out a lot of technical details that if you want to talk to me later about, we can. And trying to bring all the data to the forefront and some figures and stuff. So it's going to be a little bit less on the technical side, but more on sort of what we're doing and the types of ideas that we're thinking about and what the microbial footprint of a city might mean or how we even think about that. So this is Milwaukee, if you haven't seen that from the lakeside before. It's the Milwaukee, and this is from we have a research vessel there, the RV Niske, that we can go out on to get samples. So starting here, talk a little bit about water and why water is important, besides the fact that we drink it. So all about water. I think the Great Lakes are awesome. I think everyone would probably agree they're pretty awesome. Here are a few reasons why you might say that. Just for dollar signs and sort of public interest. They're a big deal in Wisconsin for recreational activities. It's a huge industry. It draws lots of money, both the Great Lakes and the inland lakes. Fisheries is a large thing, especially on the Great Lakes. It's a $1 billion industry. It's been declining quite a bit over the past 20, 30 years. So it's not quite as important as it once was, but more recreational fishing is sort of the bigger deal these days. Drinking water, there are 40 million people that get their drinking water from the Great Lakes, so it's a prime source of drinking water. And about half of all the water used in industry in the United States comes from the Great Lakes. So that's why what you say the Rust Belt these days is centered around the Great Lakes, is because that water provided sort of the engine to drive a lot of that industry. And what I'm showing here, just quickly, is the sort of tannish area around the Great Lakes. That's the actual watershed. So you can see Wisconsin actually doesn't have a whole lot of the watershed of the Great Lakes, only a little bit. And it actually goes just outside of Milwaukee through Wauwatosa is where that dividing line for the watershed goes. So everything to the west of there drains to the Mississippi and not to the Great Lakes. And a few more fun facts just to get you sort of engaged with what we're doing. Lake Michigan itself is the 6th largest lake in the world if you're counting by volume and you don't count the Caspian Sea, which is sort of a salty body of water. It takes about 100 years for a parcel of water to move through Lake Michigan. So when it comes out of Superior into Michigan, it takes about 100 years for that parcel of water to move back out. So it's pretty slow residence time, provides a nice system for things to grow and change. And for those who haven't really looked at or thought about bacteria and water systems, this is true for the ocean and lakes in general. You can estimate there's about 1 million bacterial cells per mil of water in any sort of inland body water or the ocean. That holds pretty true. It can go quite a bit higher in some cases. It typically doesn't go too much lower than that. There are a few places in the ocean where it's a bit smaller than that, but in general, that's what it is. And then if you take the volume of Lake Michigan itself, that would be about 4.9 quadrillion bacteria that are present in Lake Michigan. And to be honest, we don't know much about them. And so part of what I come from is that background of understanding what the bacteria are doing. So I'll say this here. And so we bring that, and now we're taking what the bacteria that are there that are doing and how we're influencing that by sort of having cities and things that discharge these water bodies. So other people are not quite as lucky to have Lake Michigan outside their door. This is just a picture of a sort of wastewater treatment system that you might see in sort of places that have less technologically advanced systems. So just a drainage ditch, and that usually dumps into a river or so on. So sewage treatment can be pretty rudimentary in places. And this brings about some big health concerns. So sanitation, which I'm showing here, so very basic sanitation sort of effort. It actually is the #1 threat to children worldwide, if you go by World Health Organization stats. So children and illnesses caused by poor sanitation is a big deal in developing countries. And as you see here, 20% of the world's population does not have access to clean drinking water, and that's only going up as sort of water supplies go down across the world. In the US, we also have problems with water. So, and a lot of it isn't as big of a deal as you see as sort of illnesses that cause mortality, but we have lots of gastrointestinal things that are related to our water quality here in the US, despite having probably arguably the best water systems in the world. They estimate that about 19.5 million illnesses per year come from drinking water sort of related things and or recreational water illnesses. More of a money-making side, around 15,000 days per year of beach closings occur. And just for an example, in Orange County, where there's lots of beaches, that can be a loss on the economy of $20 to $50 million. So water quality can have a significant impact on the resources that a community might see. And in Wisconsin, we see some relationships to these health metrics in that about 11% increase in gastrointestinal illnesses following rain events. So there's a connection between our water systems and when people are getting sick. And a couple years ago, about 10% of our beach days in Wisconsin were closed due to poor water quality. So what's this talk all about? So I gave you a little background, why I think the Great Lakes are kind of cool, how health can be impacted by water, and also sort of financial things for cities and communities related to recreational activities. But I'm coming from this microbial ecology of lakes background. So really it comes down to the who, I'm gonna say who, what, when, where, and why. So who's there, why they're there, when they're there, and what they're doing. But then that's directly tied to this microbial pollution that's coming from urban environments. So it's sort of the intersection of these two things is where this talk lies. So directly then, we're gonna talk about what are the sources of fecal pollution. So mainly the talk's gonna focus on fecal pollution, but what are the sources of fecal pollution in urban areas and how do we track them? Why do we need improved fecal pollution tracking, or do we need improved fecal pollution tracking? What's the community structure associated with our fecal pollution sources, and does that influence how and why we track some of these organisms? And then ultimately sort of this idea of an urban microbial footprint that we see in Lake Michigan. Here's a topographical map of the city of Milwaukee. So this is the city here. It basically has 3 river systems. The Milwaukee River comes from the north, the Menominee comes from the west, and then it's harder to see, but the Kinnickinnic River comes down here. They all converge in this inner harbor area into a channel that goes out, and it's probably hard to see, but there's the break walls here and here. So this is the outer harbor where a lot of the shipping industry takes place, and then that goes out into Lake Michigan. So that's sort of our basic setup, but it's a nice setup to track pollution sources coming down the rivers because they all merge into one place before going out. And so we can really hone in on what's going on coming out of the city. And so why we might do this is what it can look like coming out of Milwaukee on certain days. So if you get large rain events, this is that channel coming out of the inner Milwaukee Harbor out into the outer harbor and then out into Lake Michigan. So if you didn't think there was an interaction between the things that are out here and the things that are in here, I think this is pretty good evidence that those two things do interact on a regular basis. So if we focus in on Milwaukee specifically then, and what are the major sort of water pollution problems that Milwaukee sees? The big one that's in the news frequently, and so here's some Journal Sentinel articles, sewage treatment plants and their combined sewer overflows. So Milwaukee has a sewage treatment plant right on the inner harbor. It discharges its effluent typically into the inner harbor and that goes out. But on occasions, they can have this thing called combined sewer overflow, and this is what makes the news every time it happens actually. So what is a combined sewer overflow? If you haven't thought about sort of urban infrastructure and pipe systems, you may not have heard about this before. But basically older systems, so most of the old systems in the US, which is what is shown here, have combined sewers where stormwater and the sanitary sewer that comes out of your home both go to the same place. So they both go to the treatment plant all in one system. So what happens is when it rains, that deluge of water coming from the rain can actually flood out what they can handle at the treatment plant. So the treatment plant can't handle all the volume of water that's reaching there, and so they have to bypass their treatment systems and actually dump raw sewage into whatever receiving body that they have. And that happens quite a bit. So as you can see, these are all the communities that have those sort of systems. And the reason those were built is because practically back in the early 1900s, that made sense. You didn't have a very big community. You wanted to get rid of your sewage, so the best way to do that is to dilute it, and a good way of diluting it is by rainwater that's coming in. And so that's why they were built. Since then, they're typically separated now, so you have one system for stormwater, one system for sanitary sewers. And so that doesn't happen. You don't get these overwhelming, well, in theory anyway, that doesn't happen. You don't get overwhelming sort of rainfall events that flood the system. So that's what it shows here. It bypasses, they have a bypass and different lines in their system, and it goes into basically into the river systems in Milwaukee, downstream a little bit, and then into the harbor and out. As you can see, this is a pretty big problem in the US. There's current estimates about 900 billion gallons of raw sewage, which includes stormwater, gets discharged out into the waterways. And there's something around, like it says here, about 9,300 of these sort of sewer overflow places in the United States. In Milwaukee, this actually happens only about 3 times or so per year, maybe on average. Other cities, it can happen quite a bit more. There are several other communities that don't have as good of a system as Milwaukee currently has, and so that they are almost constantly discharging sewage out into the environment. Just to give a plug for sort of the Milwaukee Sewage Department, and if you want information about sewage treatment and what you can do to help your sewage treatment plant deal with water issues, the Milwaukee Metropolitan Sewerage District actually has a really, really nice website. It's really easy to navigate. It's full of data about their system. You can go there at any time and see what their capacity is at that moment, what they're treating. So if there's a overflow coming, you can actually kind of predict it from what's going on. And then, so that they have all that information, including information on like rain gardens and rain barrels and things that sort of help prevent these sort of things from happening. And they took a pretty progressive step, I guess, in the late or early 90s. This used to be the amount of sewage overflows that Milwaukee had. So we're up here in the billions of gallons range, so 7, 8, 9 billions of gallons per year. What they did was they built a deep tunnel. So they dug a huge pipe in the ground that essentially can store water. So when we get these huge rain events, they're able to shift some of the water that's supposed to be coming to the treatment plant, put it into the storage container, let it sit there, and then as the rain subsides, then slowly treat it and send it out. So because of that, you can see the reduction in amount of overflows that they've had, and the volume has gone drastically down in that time since then. So that we don't have nearly as many in Milwaukee as they used to, and I guess their data is about 98% of all water is now currently treated. So they're doing a pretty good job, and this came as sort of a big funding initiative by Milwaukee and the federal government to clean up sewer overflows in the United States. However, that's the big one, the one that gets all the news, but it's probably not the one that makes as big of a deal, or is as big of a deal in cities like Milwaukee. It's more the second thing, failing urban infrastructure. So just like building that deep tunnel helped alleviate a problem that was predicated by it being built in the early 1900s, most of our pipes in these systems were built in the early 1900s and so on, and they only had 50 to 100 year ratings on how long they should last. And so a lot of the pipes are starting to fail, and they're having problems sort of getting the water to where it's supposed to go. And it's difficult to fix them because you have to tear up all the streets and tear up everything. And so it's not a particularly ideal scenario for people living in neighborhoods to have their street torn up for a year to get pipes fixed. So most people just ignore it. But here's the problem. So we have old cities and old sewer pipes. Just as a bit of a tangent, this separated stuff, so the stormwater that we have, emits about 10 trillion gallons of water into waterways. So we also have, besides the sanitary stuff, which is in the billions of waters in overflow, we also have whatever runs off the land gives about 10 trillion gallons of water into waterways. So this can also be a problem and is related to this. Because we have all this water going out, often we get water that doesn't make it to the sewer district. It is going somewhere else, and frequently it's ending up in the stormwater system. So estimates are about 30% of all sewage doesn't actually make it to the wastewater treatment plant. So if you can imagine that, a lot of it is leaking out of the pipes into the ground. There's been a lot of deal over sinkholes lately, has been coming up in the news a lot. Some of that is due to this water is leaking into the ground and causing erosion of the soil underneath places and so on. So that's part of that problem. But also it can get diverted into stormwater systems and so on. There are old connections that were built in the 1800s that they don't know about and so on. So there's all sorts of issues. And they're estimating that this isn't going away because for the money they need to fix this compared to what they're getting, it's just getting worse. And so in the next 10 years, they're guessing they're about $100 billion short of having enough money to fix what they need to fix. So it's only going to only become a problem that's going to become worse. So we have this thing, which is sort of we know when this occurs and when it happens. This is hard to track because it can occur anytime, and we don't know where they are, and often it's difficult to pinpoint. And then we have a third sort of scenario of bringing fecal pollution is urban animals. So You know, there are dogs and gulls and things on beaches or in waterways. They all poop, they all defecate in the area, so they also bring sort of fecal contamination. And some of this stuff their fecal contamination may not be as important in some cases because the health risk for humans associated with their fecal contents isn't typically as great as stuff that comes from sewage where it's loaded with human viruses and stuff. And so separating out these sort of sewage issues from these issues can be a big deal, and understanding what's going on in the system and pinpointing where you want to target remediation efforts. So human health risks from fecal pollution. As I mentioned, there's a lot of viruses in sort of fecal pollution that goes out. That cause problems, and so we want to know, how do we track these? We really want to track the pathogens. The pathogens are mainly viruses, but the problem is the viruses are rare, they're difficult to culture out and or identify. They're really patchy, and and even though they're rare and patchy, a single virus can cause an infection. And so they're difficult to detect, but they're there, and their low numbers is significant, and so that causes issues. And there are lots of them, so it's like, which ones do you detect? Which ones are the markers? So one way is to actually track the pathogens, but it's technically more difficult. And so many years ago, they came up with this indicator paradigm where we know there are other organisms that are associated with fecal material or other materials, and where the viruses are. And so if we track those organisms which are more abundant and easier to get out, it'll be easier to track the pollution than trying to track the viruses themselves. And so that's where this came from. It's called, you often call it fecal indicator or pollution indicators. And as I said, they're more abundant. It'd be ideal if they covaried with those pathogens that are in the water. And you can track a single group instead of having to track many. And there are all sorts of tests that have been developed over the last 50 to 100 years actually to do these sorts of things. And current recommendations, so the EPA has recommendations for beach managers or water quality managers on what they should look at. Related to E. coli and enterococci are typically the two that they're still recommending. So these are culture-based methods where you culture out E. coli and enterococci on some selective media. It tells you what the concentration of those organisms are, and based on some back calculations for what we know is in sort of infectious doses of the viruses as related to these indicators, we can figure out how many of these could be a problem if you detect them in the environment. So they have actual parameters. If you detect, let's say, 30 of these per 100 mils in your sample, then that's a cause for concern, and an advisory has to be given. So that's how that's done. And ideally, we'd want these to be abundant, right? So these organisms, to give better sensitivity, you'd want them to be particularly abundant in the sources that you're trying to track. Also, another thing is if you want source specific, so if you want to know where to go in your system to mitigate the problems that are occurring, you want to know, is it sewage that's causing the problem, or is it stormwater that might be carrying sewage, or is it stormwater that might be carrying animal pollution, or is it just that you have lots of gulls on your beach and they carry E. coli and they're defecating on your beach, and that's a problem, but not as big of a deal perhaps if you're getting sewage. And so ideally you'd want to know where your pollution sources are coming from so that you could target the money that you have to fix those problems. You'd want them to be common, so if they're associated with either sewage or particular organisms, you want them to be commonly in those organisms, not a patchy distribution. And you'd prefer that they didn't grow in nature. So once they're released, you don't want them to persist. So you don't want to be detecting E. coli that's in sand that was deposited there a year ago, because it has no relevance on the sort of health implications of what's there. And so all of these 4 things are what we'd like to see from indicator organisms. Unfortunately for E. coli and enterococci, the only thing they really are common in organisms. They're not actually particularly abundant if we're talking about sewage-related things, and we'll get to that in a second. They are really abundant in some animal sources. They're definitely not source-specific. Almost all mammals have these in their guts. And they definitely have been shown that they can grow in nature, and there are actually some E. coli strains that are naturalized sort of soil beach organisms, and they actually look very different than the E. coli that you have in your gut. They're not commensal gut organisms. And so that's also a problem, is are you detecting something that's coming from someone, or are you just detecting something that naturally lives there? So all these issues have been raised. And so it's how do we get around this? This is what we're starting to identify as a problem, and can we do better? And the can we do better really came about recently. And there's sort of a paradigm shift in understanding microbial communities in nature. And this all became because of sequencing technologies. So sequencing technologies have changed radically. Well, they're changing radically every year, maybe every 6 months. But over the last 10 years, they've changed dramatically and have altered our ways of, and our perception of what microbial communities are and what they look like and their complexity and nature. And so the basic idea for tracking organisms and identifying bacteria from nature without culturing them is this sort of process here. And this is a really simple schematic, but you have bacteria that you collect, either they're in soil or you collect some water on a filter, something like that. You extract the DNA from these bacteria, so you pull out all the DNA from this community. You then amplify for a specific gene, and I won't go into this, but this gene is used to taxonomically classify bacteria. Just like you would look at the morphological characteristics of birds to tell you what bird species they are, we use a specific gene to sort of tell us what bacterial species it is. From there, you amplify this gene, and then using that amplified pool, you can then get the DNA sequence of this particular gene, and based on gene-to-gene comparisons of all these different organisms that we've pulled out over the years, we can begin to identify what's there and what the diversity looks like and so on. So that's the basic process. And for many years, doing this process required a few steps that allowed you to pull out a few organisms from the environment. So a big study looking at environment, you might be able to get 100 to 1,000 sequences of bacteria. That was a pretty significant effort even when Greg and I were starting in graduate school, not all that long ago. And so that has gone from this sort of technology, so 100 to 1,000 was a big deal in understanding the environment. And remember, there are 1 million cells per mil of water, and there's probably thousands to tens of thousands of actual species in that mil of water. So we're just scratching the surface of what's there. Along came these next generation sequencing technologies, and I'm trying to think, it's probably been 6 to 7 years now that they've been more widely applied, but have really taken off in the last 4 or 5 years. And this was, as I say here, a game changer. And the one that was really took hold first was a technology called 454 sequencing. They were originally a company on their own, and they got bought out by Roche. But essentially what happens is, instead of sequencing just a handful of things at a time, they use this bead technology to capture single strands of DNA. And these beads are really, really tiny. They can then take these beads, separate them into these tiny, tiny pores. So you can do, instead of doing 100 of these, you're now doing millions of these. And so by doing this sort of amplification procedure in these little tiny reaction wells, we've gone from getting 100 to 1,000 sequences per sample that you're interested in to now tens of thousands is really common, and some newer things you can get millions pretty easy. So we're getting really a much deeper look into the microbial community than we ever have before. And because of that, I think the big thing that people may have heard about, and I've heard that you've talked about this a little bit, is the human microbiome. So there's a big research effort spearheaded by NIH and some other groups in Europe to understand the bacteria of the human body. And this sequencing technology that I'm talking about here has really been driving this research. So these are just a few papers, and these are actually pretty old now. They're 3 or 4 years old. There's hundreds more since then. And it's all, or not all, but much of it is based on deep sequencing of microbial communities to understand who's there, so then you can start to generate hypotheses about what they're doing and what they are now that you know who the players are in the system. So we saw that. The PI that I work with, Sandra McClellan, saw this about 4 or 5 years ago and said, oh, that's interesting. I think we can take advantage of that. So on the next slide, we'll get to how we did this. But before we do that, I'll take one last step back here in sort of the background of my talk. And I just want to talk about a quick statistical technique or a visualization of data technique. So when you're generating millions of data points, essentially, so here's a taxonomic assignment of some bacteria. You can see each one of these columns, let's say, represents a sample. The numbers in the columns represent the number of organisms or sequences in that sample for that particular organism. So you're talking about big data here now. So it's how do you visualize or look at that data? And just one technique that people use is called ordination, or principles based on sort of ordinating things. So let's say you have this set up. You have these different samples. There's 7 samples. You compare those samples based on the distribution of these organisms. So each one of these samples has this distribution. You can compare those distributions to each other using very different statistical tests, and you get a number that says a similarity. So in this case, sample 1 and 2 are 90% similar in their composition, and so on. You get a matrix like that. That matrix can then be represented in two-dimensional space. So you can say these two are the most similar. They're 90% similar, so they get put close together. Two things that are not very similar get put very far apart. And so then you can say visually, these communities are similar to each other and more similar to each other than they are to the things over here. That's one way of doing it. You could then add some sort of metadata parameters based on when you took the sample or so on, and start to ask questions. Do you see a seasonal progression in your community and so on? And you can do tests on these patterns in your data set to get at those answers. So this is one sort of plot that I'll be showing a few of these that kind of look like this, sort of. And then another way is a dendrogram. This is kind of a more common way that you might see this. So relationships based on sort of connections, again, based on the similarity of the communities, not the phylogeny of the organisms as you'll frequently see, but these are actually community similarities. So those two ways you'll see a couple times in the talk. Okay, so let's go to the data finally. So interpreting the human microbiome. So there's obviously lots of interpretations, but this data is out there. And it's mostly, once it's published, it's freely available to the public. And so we have access to these data. And so we started looking, well, if we were going to look for human fecal pollution, why don't we go and see what's in human feces? And so that's what this is representing. So these are just two pie charts of what's the order classification of bacteria. So it's a really broad grouping of bacteria. But I wanted to point out, these two things are from around 200 different human fecal samples. And one of the big things the human microbiome has found is that obesity and microbial community composition are related. They're not sure what direction it goes, or if it goes both directions and that one causes the other and so on. It looks like there's an interplay, and they kind of both can contribute to each other. So your community can actually affect your sort of fat retention and things, and what you eat can affect the community. And so there's this interplay. So we have these two different sort of human groups. One group is dominated by this group of Bacteroidales, and one group's dominated by Clostridiales. But you can see the majority of sequences, the majority of bacteria that are present in the human gut are from these two groups. So if you were going to design things that target fecal pollution, you probably want to go after one of these groups. And E. coli and Enterococci are not part of these groups. So they're probably not the dominant members of things that if you want something that's abundant, it's not something that you want to be looking at. And so I said that here, these are really abundant. This particular group here has never been explored as sort of indicators of fecal pollution. This group has a bit. And in general, in human feces, E. coli and Enterococci are both less than 0.1% of the community, maybe even lower than that I have there. So we said, great, we have data from the human side of things, but we don't necessarily track directly human fecal pollution. We're tracking it after it goes to the sewage treatment plant, or as it's being released through a pipe system. So we said, let's look at sewage. No one has ever actually looked at sewage in depth for its microbial community. So this is sewage influent. Once it's been collected from all the households in Milwaukee, it comes to us in a single influent into the treatment plant. And so at that influent point, we take the samples. This is a really complicated bar chart of the, you could say, sequence types or microbial species, if you will, that are present in these samples. This is Milwaukee sewage influent taken over a 4-year period, and we take it about a month every year. And so we have right here 41 samples over this 4-year period. One thing to notice is that it's remarkably pretty consistent, and so I'll get to that in a second. But other things to notice I want to point out, there are some groups that are quite abundant in the sequence libraries, and I point out some of their names here: Acinetobacter, Aeromonas, Archobacter, and so on. Those two groups of fecal bacteria that are really abundant in human feces are represented by this barred area here. It's hard to see which ones they are, so I just put a rough area of how much. But as you can see, that's actually not very much of this community, right? I mean, if most of the sewage is fecal material, then you'd expect these to be quite large because they make up 90% or more of the fecal material coming from humans. So actually what we have is quite a large amount of or potentially non-fecal bacteria. And just to reiterate, ignore this part right here because we'll come back to that later, but E. coli and Enterococci in sewage, again, less than 1%. typically, and so E. coli especially low. So again, probably saying that your detection limit is not going to be very good if you're looking at these things to look at sewage. So I just said that probably the fecal content is not very high in sewage, or the sewage microbial community. And so then we said, okay, let's look at this a little closer. And we picked out those 3 groups of bacteria that are particularly abundant in the sewage influent: Acinetobacter, Archobacter, and Trichococcus. You can see here's their average abundance, if you will, over the sewage samples. So they range somewhere between 20 and 30% of all the sequences that we recover from those groups. Here are typical, what we know are typically fecal bacterial groups. So these are pretty broad groups. So they range somewhere between 10 and 15% of the community on average. And these are just by saying these taxa we know are primarily fecal taxa, and so we're just going to call them fecal taxa here. So this is our sewage influent. If you compare that to a human, note the scale difference here. There's occasionally some of these groups will show up at a very, very low level. So we're talking less than 0.01% of the community, in some cases for Acinetobacter. But the human, these groups here in a human fecal sample make up 90% or more. And so more evidence that we're probably, the majority of bacteria in sewage is not fecal related. So that's important because if we're going to track this, there's 2 things that we can get at. If we want to track sewage, maybe this is what we want to track. Even though they're not related to fecal content, this might be a good marker of sewage distribution in your system. But if we want to track fecal bacteria, we've got to separate out which one is which so that we know that we're tracking fecal bacteria and not just pipe bacteria, if you will. So let me ask questions. Well, then how similar are the fecal bacteria that we're seeing in sewage to what we see in humans? And this is a heat map. So the colors here represent sort of relative abundances in our samples. So red is up to greater than 3% of the samples is represented by this phylotype of bacteria. So you could count this as a bacterial species if you wanted to. You can see sewage is in the left column here. Humans are in the right column. And it actually tracks pretty well. Things that tend to be really abundant in our sewage samples also tend to be quite abundant in human fecal samples on average. And so what that is telling us, and this I guess I should point out, the sewage samples are from Milwaukee and these human samples are from Missouri and San Francisco. So we're not talking about people that live in the same area as what we're actually tracking in Milwaukee. So what this leads us to believe then is that sewage is really, as you might expect, first thinking about it, is really a collection of the population of humans. So we're seeing the most abundant things in humans on average, even though humans, as been pointed out in the literature, are quite different from person to person. So there's large variations in your gut and fecal content when you're looking at each individual person. But when you put all those people together and they're flowing their fecal content to the sewage treatment plant, it turns out that that averages out, and you're actually looking at sort of like a population or demographic of your city, the microbial community of that city, essentially. And so that was a good sign for us. So what we're seeing in sewage is trackable back to humans, and we have this sort of non-fecal component that is also quite interesting. Again, one more look at this. This is a specific group of bacteria called Lagnospiraceae. It's a family. This is the prevalence. There's 48 human samples that we were looking at here. This is the prevalence, so the number of humans that they occur in. So like this sample, these samples down here didn't occur in any humans. These particular sequence types, these occurred in 48. And this is a relative abundance in sewage. So ignore these numbers. They've been sort of normalized between samples. But essentially, as you get more prevalent, so things that are more common in across humans tend to be more abundant in our sewage. So just reiterating the fact that we're seeing a population of humans, that's the microbial community that we get in the sewage influent. So the next maybe potentially obvious question is, but are my city's microbes like your city's microbes for sewage? And so we asked that question in a simple little study. We took 12 different sewage treatment plants from around the US, compared their microbial communities. I'm showing another bar chart here. It's kind of messy again. But just to show that if you look at the colors, they're relatively similar across the different places. And look at that a little bit more in depth. This may be difficult to read, but these 2 groups here that I've circled, so there's 70. These are 70 taxa of bacteria and 83. These are actually sequence types that we identify in these systems. These were found in every one of these systems, so all 12. So there were 100 and there are more than this, but these were particularly interesting to us. But these 150 or so bacteria made up 60 plus 10, so 70% of the community of all the things that we recovered were shared across every site that we looked at, from Hawaii to Florida to Maine and so on. So it says that whatever system that we're looking at, this sort of sewage pipe system, is some sort of environment that is similar across geographic differences with temperature differences and demographic people differences. So we're, are we getting a community that is a pipe sewer community? So that's something that's different and not a lot of people think about is a pipe system environment that could be studied. Moving forward, this is ongoing research. We've expanded our 12 cities to 80 cities across the US, and we're also doing some temporal dynamics here, so across time. And so we have 80 or so sewage treatment plants enrolled, and these are the different people that have agreed to help us, and they're sending us samples to look at how these things vary in time and space across the US. So remember, I said non-fecal bacteria are the majority. So quick note, remember that again. So to look at that, but the way we did that previously was we just said, here we see these bacteria, they're in the sewer pipes, but they're not in humans particularly, and we know that these groups of bacteria tend to be associated with fecal content and so on, but it's not really terribly statistically sound. And so we used a program that was developed just a couple years ago by this guy, Dan Knights, and he uses Bayesian inference to classify bacteria to their source. So if you go to a community out in the environment, what he's calling a sink community. So your sink, you can then get a distribution of the bacteria in those samples from that community over time, or space, or whatever. Go back to communities that you think could be contaminating that community, sequence in those communities, and then use Bayesian statistics to tell you what proportion of your sink community might be made up of these source communities. So it's quite an elegant sort of technique. It still needs some refining and so on, and we've been in discussions a little bit with him about that. But it gives us a sort of statistical estimate of those groups. So again, what we're calling fecal bacteria versus these sewage infrastructure bacteria, and then also transient, or things that weren't associated with enough treatment plants with our depth of sequencing that we couldn't tell with certainty with our statistical measure that they would be a sewage infrastructure bacteria. So you can see the fecal content of these places, we're estimating around 5 to 20% of the bacteria coming in, or of the sequences that we get, and the sewage is again about 70 to 80%. And there's a variable sort of range of 5% or so of things we can't classify. So just backing up what we saw before with some stats to show that we think that these are non-fecal bacteria. Again, thinking about pipe communities as a system, I show this just to show that we actually see temporal dynamics in our pipes in Milwaukee. So this is 2 sequences. So these are 2 sequence types of Acinetobacter, a genus of bacteria. They actually differ by only a couple base pairs in their sequence, but one of them tends to occur in more abundance in the summer months and then declines. So look at the filled circle, so it has this sort of temporal dynamics. The other one has the exact opposite dynamics. And so we are actually seeing sort of ecological temporal patterns in these pipes. So again, more evidence that maybe we're really looking at a sort of modernized or human-created ecosystem for bacteria that we haven't really looked at or thought about a lot previously. This is essentially one of those ordination techniques that I was talking about, where we're putting samples that are close together. Things that are close together are more similar than things that are far apart. And the outer colored lines and things represent taxa that are associated with those particular samples. Here we divided up our little 12-city study into sort of northern and southern latitude because we were interested in whether there was differences in the temperature, because these samples were taken at kind of in the cooler time of the year, and so we were wondering if there were temperature differences that affected the communities that we're seeing. And in the fecal community, not really. We just saw all the communities were pretty similar once we divided those out. When we divided out the non-fecal bacteria, we actually did see a bit of a pattern in that the northern latitude cities, minus this one city, were typically more similar to each other than the southern latitude cities were more typically associated with each other. So there might be also sort of environmental parameters that are actually affecting the non-fecal, or what we're calling non-fecal component of these pipe communities. Okay, so just to close on the sewage part of this talk, and then we'll move back to the lake here in a second. Remind you that I think that they're likely the non-fecal bacteria, the majority of the organisms that are in sewage. Sewage is really a reflection of the human population, so we have a nice sort of demographic thing that you could look at. Traditional fecal indicators are not particularly abundant in sewage, so may not be good indicators of human fecal pollution. The microbial community composition, I would argue, is surprisingly consistent across these different treatment plant influence, and that we're really looking at probably a unique ecosystem that may have unique dynamics as organisms pass through there and could affect either treatment plant operational conditions or how we view these communities and how they change as they come out of your house and into the sewage treatment. So next, we're starting to look at what are the origin of these non-fecal components. We really don't know where they're coming from necessarily. Could we use sewage microbial communities to look at human demographics? So sort of a health sciences approach. Do the communities that we're seeing and the parameters associated with human health that we see in individual humans relate at the sort of population levels for cities? And then, of course, the real practical part of this, developing new assays to track pollution as we move out into the lake. So now we're going out, actually almost going out to the lake. I said in the beginning part of the talk, sewage is not the only thing that causes sort of fecal pollution problems in Milwaukee or in other places. The other two big things are stormwater. Here's another heat map with abundances. So red is really abundant, blue is non-existent in our samples. And so this is a sewage sample, in particular taxa or sequences associated or each one of these lines. Here's stormwater. You can see some of the things are shared between sewage and stormwater. And that could either be that they're pipe bacteria or that there are some sewage in your stormwater. But there's also this huge component down here, and all these groups are typical fecal bacteria. So they're found almost exclusively in sort of human or animal guts. So we have this huge amount of things that are we've never seen in our sewage samples. So either they're really low abundance in that sewage sample, or they're not human affiliated. So it suggests that there are other sources of our fecal pollution in our system. And we guess that they're probably animals, so we've begun looking at animals, and I'm not going to go into this because this is some other people's research, but here's another one of those plots. These are fecal samples taken from various animals and compared. And so you can see humans are here, dogs and cats cluster together here. So you can see sort of the relationship that some animals have communities that are more similar to humans than others, like humans and cows are pretty different. So ruminant organisms look quite different than humans and so on. So we're beginning to look at, and this is a particular visualization technique that I'll put a plug in for Murat Aaron at the Marine Biological Laboratory is sort of working on developing. And it's quite a nice way to look at these big data sets. Okay, so finally, we're going back to Lake Michigan. These are our sort of common sample sites that we routinely monitor when there's sewage overflows or go out at various times of the year. So you can see we go all the way, our institute is actually right here. So we go up, out the river, out the gap here into the harbor, and then out into the lake. Just a plug, since I'm a microbial ecologist at heart, here's what a bacterial community from the lake looks like. These are aggregated into fairly large taxonomic groupings, so that's why there's not very many. Probably the most abundant freshwater bacteria on the planet comes from this group here called AC1. Has no cultured representative, so we don't have a name for it, but it has a nomenclature called AC1. It's found in every freshwater body that's been sampled in the world. And there are some differences among it, but it is quite common, and there's finally some genomes and things starting to come out from some sort of novel ways to pull out single cells from the environment that are giving us insight into what it might be doing and why it's so abundant in lakes. So I'll just put a plug, and if you're curious about sort of freshwater bacteria, this is a paper of mine, so plugging myself, shameless plug for myself, called The Guide to the Natural History of Freshwater Lake Bacteria a couple years ago, that if you're curious about what's in freshwater, I would suggest this is the place to go. However, we want to know, well, if we have these sewage communities, do sewage communities look like lake communities? I mean, I probably already knew that answer, but it's a question that you have to ask. Here's a dendrogram showing all the different communities that we've sampled and their relationship to each other. So branching back, here's a few lake samples here. Here's sewage here. They branch back to here in their relationship. So they're not very similar to each other, community composition-wise. They're more similar to each other than they are to, these are fecal communities, so because of the water component that's in sewage. So they're different, but more similar than the fecal components that we look at. If we track then some of these organisms that we are interested in, these same groups, the sewer-affiliated bacteria, Acinetobacter, Archobacter, Trichococcus, and these fecal families out in the lake. So here's a big combined sewer overflow. They're making up 3% of the bacteria in the harbor at that time, the sequences we were getting. And as you went out into the lake, they declined. This is another sort of sewage overflow event where we had the rivers and they were quite abundant there. And it declined as you went out in the lake. Here's a lake where we're getting a day where we have no rain events associated. We did have some low level amounts, and we know that Milwaukee's rivers and harbors pretty much have a consistent sort of fecal signature presence in them, and probably are getting some chronic sewage delivering from pipe aging and stuff like that that's coming out of the stormwater system. But so as you can see, it looks like looking at these sequences, some of these groups might be decent to track or use as new markers. We've been working on assays to develop these. Here's another one of these crazy plots that shows relationships. All the communities are these little gray dots that are hard to see, but sewage is in green, human is in blue, and cattle are in orange, because we're interested in this signal as well, because they're upstream in Milwaukee. There are some large cattle feedlots that can contribute to the Milwaukee waterway pollution and makes it all the way down to Milwaukee. So we're interested in what we see out in nature. And so looking at this plot, you can see there's some organisms that are over here that were not found in the cows, but were found in the human and in our sewage. And we targeted this one group that we call LACNO2 to design assays over. And that's what this is showing. This is the relative, or the abundance of this group in our sewage samples as compared to E. coli and enterococci. So it's a bit more abundant than they are. And this is a single sequence, so it's much actually less, it's actually even probably less robust group than these two. These are bigger, what I'm trying to say, taxonomically larger groups than this, so this is even more refined. So the fact that it's more abundant is good. And it's related to this organism, Bladia. So it appears to be some sort of Bladia-related organism, which is a new genus that was put forth about the time that the human microbiome stuff started. If we actually track this, so we develop qPCR assays for this particular marker. We track this under different scenarios out in Lake Michigan. So here's a sort of heat map, look at it, and the copies per liter of water that we're seeing of these organisms. Excuse me. In sort of dry weather, this is an average of 4 or 5 events. You can see there's some low-level detection of this particular marker in the river area, harbor area, but then as you get on the lake, it pretty much disappears. When you get heavy rains, that gets to be it goes out even a little further, and then when you get these large CSO events, you can get huge concentrations of this organism, or the sequence of this organism out into the lake. Even out to 5 miles, we can detect it pretty readily. So even though it's been being diluted out at a pretty high rate as it moves out of the system, we see pretty strong signals. So it appears to be a reasonable marker for looking at our sewage pollution in our system anyway. And as it turns out, it matches better to sort of the human health risk part of this, the virus content, than some of the other things like E. coli and enterococci. Here's a plot of the copies of this per liter versus adenovirus copies per liter in our samples. As you can see, it's pretty patchy, but running a logistic regression on this, as we increase the concentration of our marker detection at tenfold, we get about a 154% increase in the probability that the adenovirus will be present. So it is correlating to adenovirus concentration, just as you imagine, the ecology of virus release versus bacterial release out into the environment don't always match up perfectly. So we're seeing sort of likely ecological effects as they get released, which we're trying to understand. Also, adenovirus is one of the best viruses to study because it's pretty prevalent in human fecal content, but it does also have seasonal cycles, so it goes up and down and things, and so it sometimes doesn't appear even though there may be sewage present. So one of the last slides here, we have, if we extend this source tracker classification method, we can look at other communities. So we have sequence communities from, as we said, fecal stuff, humans and cows and things, sewage. We have some soil samples, stormwater samples, river, lake, and so on. If we use this to classify all the different sequence types that we're getting, we can get a statistical estimate of the number of bacteria that are coming from each of these different sources. You can see this is a transect during a big sewer overflow from the harbor out to 5 miles. And as you might expect, what I'm calling sort of the urban signal here, the fecal and the soil and the stormwater, they're all being washed out together. It's pretty large when we have this huge rain event, and then as you go out, it decreases. You can sort of see the utility of this here is visually like in a plume. So you can, this is a huge CSO, so all the way out to 3.5 miles from this point here, you could still visually see this plume. And so you can see the difference here when you just move, we're at pretty much the same point, just moving in and out of the plume with a boat. You can actually see the drop in those bacteria. So it's sort of a truth check on whether this is working, but it seems to be working reasonably well. So it's something that we're looking into more in the future is, can we use this to identify where some of these bacteria are coming from? And I didn't mention this, but real quick, this technique was actually developed as part of a grant for NASA, because they have these clean rooms and they want to go to outer space and try to collect bacteria. They want to make sure that they have no contamination before they go out. And so they used this, or he developed this for them so that they could look at where the pollutants that they were detecting in their clean room might be coming from. Just aside on that. And then if we look at, as you might expect, rain events, so this is that classification of urban associated bacteria. It actually correlates pretty well with the amount of rain that we get over time. So more rain, we get more of these urban associated bacteria and this is in the harbor. So it appears that this classification thing is at least working to the point that it might be useful. Probably needs some refinement. But so to wrap everything up then, I will say on the Milwaukee's microbial footprint, I think we're getting to the point where we can track both fecal and non-fecal sewage bacteria out in the environment, both with sequences or PCR-based methods. And we're working on developing sort of some techniques that we could do this in more real time, either with sensors out in the environment or quick sort of new techniques to extract DNA and on-site detect these things. We can find urban-associated bacteria out to 5 miles from shore, so the area, the microbial shadow, if you will, of the city, it can be quite large at times. And it's all associated with rain events and sort of this flow of water coming out of the city. And you can see here, I pointed out, here's where you can see the plume coming out, and there's clean water here versus really dirty water coming out of the city. Oops, hope I go the right way. So finally, what we're looking to do in the future then is to combine some of our assays to get sort of these source profiles out in the lake, begin relating the discharge that we see coming from the harbor to beach health. So the beaches in Milwaukee are a little bit away, but how much this actually affects these beaches. We study quite a bit, but we don't have a great handle on that. But particularly, it turns out that it seems like the outfalls that are near the beach are a bigger deal than actually this water coming out of the harbor. We're looking at persistence and decay, so ecology of sort of the markers as they go out. Do some of them persist longer in the environment than others, and what does that mean for when we're detecting them? Or some of them rapidly decay. The particular organisms that are associated with human fecal contamination are usually strict anaerobes or fairly strict anaerobes, so you might expect them to sort of die off or decay quickly. Whether or not that's happening, we're not sure, whereas E. coli and enterococci are not complete anaerobes or strict anaerobes, and so they can actually live out in the environment more readily because they can use oxygen to grow or to maintain respiration. And then finally, are these, combining my interest in lake ecology and these bacteria, are these urban associated bacteria active? So once they are discharged out in the lake, do we actually see activity? Are they contributing to anything that we might measure for ecosystem function-wise, whether it be nitrogen and or phosphorus cycling or respiration or measurements of that, so that we can get a better handle on nearshore environment, sort of fluxes to the atmosphere versus offshore stuff. So that's also another part that I'm looking at a bit. So with that, I'll wrap up. I think we're right on time here almost. And acknowledge all the people. So Sandra is the PI in the lab that I'm associated with, and all the people that do the work. We have funding from a couple of places, and then collaborators, especially at the Marine Biological Laboratory, that do the majority of our sequencing, and then our ship captain, Greg, and his first mate sort of that take us out when we go out in the Niske. So that, I thank you for your attention, and I will take any questions when you get them.